Jason.
Thank you. And thanks for inviting me to participate in this boot camp. What I plan to do is give a very, very basic introduction to thalamus and cortex. And in fact, the outline for the talk will start off with very basic understanding or description of the properties of thalamus. And then I will talk about a division of thalamus into core matrix. I won't have a lot to say about this, but I think it's important to bring this up, at least initially. I'd like to talk about the circuitry within thalamus and some of the synaptic properties that are involved. Then switch to cortex. And again, this is going to be a very brief, superficial view of cortex, at least in the beginning. Cell types, layers and columns in cortex. Area specializations in cortex. And then I'll switch to the relationship between cortex and thalamus and a feedback, layer 6, Cortical Organization. This is a very poorly understood pathway. It's a massive pathway, and I'll try to explain why it's so difficult to study. And then if time permitting, and I'm very flexible about timing here, I'll get into more complicated stuff. I think it goes without saying that you should feel free to interrupt me at any time. And I'm going to ask you to be patient because I don't hear very well. So I may have to ask you to speak up or repeat, but we'll get through that. Okay, to start with thalamus, this would be an early embryonic view of the brain through the third ventricle. And this has the region where thalamus is going to evolve from. There's a region called the epithalamus, which is not of particular interest here, but this is what's known as the dorsal thalamus because it's found more dorsally, and that's the reason for the name. And this is where all the relay nuclei exist. That is, this is where all the regions, all the cell groups that project to cortex are going to develop from. But there's also ventral thalamus, which includes the thalamic reticular nucleus, a region of the lateral geniculate that is really not going to be much important for what I have to say, and sometimes a region called the zona incerta is included there. But the main area of ventral thalamus that's important is the thalamic reticular nucleus. Now, to put this in context, the thalamus is a paired structure right at the midline, right in the middle of your brain. And each one of these is about the size of a walnut. So again, this is a view of the brain. Lateral surface, medial surface, this is the thalamus. And these are the sort of a cartoon view of the thalamus. This would be the left thalamus back to front. And this shows the dorsal thalamus with all the relay nuclei. And this would be the thalamic reticular nucleus, which sits sort of as a shield up against the lateral surface. It's pulled away and cut off so you can see the relay nuclei. So this is where the cells of projector cortex live. The thalamic reticular nucleus does not have cells of projector cortex. Instead, all of these cells are inhibitory. They're GABAergic. And they project into the main nuclei, the relay nuclei, to innervate relay cells and inhibit them, provide powerful inhibition of relay cells. Now, a couple of additional points here. One is that all cortical areas, as far as we know, receive a thalamic input. This is a general property of thalamus and cortex. And second, virtually all information reaching cortex must pass through thalamus. So this is a very strategically located and organized structure. Basically, information from the periphery that comes in has a bottleneck through thalamus. And we think thalamus plays a crucial role in things like attention, meaning that it can block the flow of information, particularly if you're not attending to it. And one of the roles of thalamus is to act as a gate, to control what information reaches cortex and what doesn't. Yes?
Does that include other cortical inputs, like from other areas of cortex?
To do what?
Passing through the thalamus?
Yes, I'll get to that. If I don't, ask the question towards the end again.
I'm wondering if there's an external mic. I
Sorry?
think your voice is a little bit soft, so it might be better if there was a mic. Should be.
See if we can try a different place here. Is that better?
Maybe it's okay, but it just seems just a little bit more soft. So there was a mic.
Can you turn the volume of the mic up back there?
I think it's getting better.
I'll try to speak up. I'll speak louder. Okay, so what I want to spend just a few minutes is talk about the core matrix division of thalamus. These terms won't mean much until I talk about them. If we make cuts through the brain in this in what's called a coronal plane, you can see thalamic nuclei here. The ones outlined in green are part of the core thalamocortical system, and there are ones in white that aren't. And a feature of core pathways is they project each one of these nuclei to a relatively limited region of cortex and mostly innervate the middle layers. The matrix cells are located throughout thalamus, concentrated in these red zones, but found in every single thalamic nucleus. So you'll notice that what we call a core nucleus also has a smattering of matrix cells. These cells are very diffusely projecting. Instead of being topographic, they project very widely through cortex, and they mostly innervate layer 1. Another feature of matrix system is that these cells also innervate the basal ganglia and amygdala. These innervate only cortex. So it's not known whether individual cells branch to do both or whether they're separate populations. And in fact, one of the problems with the matrix is that although we think it's a very important system, this is about all we know about it. It's thought to play some general role in overall arousal. It used to be called the reticular activating system. But frankly, we know very little about the cell properties that give rise to it and the synaptic properties up in cortex, because innervating layer 1 provides a very difficult system to study. And when I talk about the layering of cortex, I may come back to that. This is all I'm going to have to say about matrix. I'm just alerting you that these pathways do exist. I'm sure they're important. And the frustration is, even though they were identified in the 1950s, we haven't advanced our knowledge of them very much at all. So I'm going to concentrate on the core pathways that we know a lot about and talk about basic circuitry there. Are you able to hear me in the back?
Yes.
Okay. So this is a section through the thalamus, through the somatosensory thalamus in this example. And this is the TRN more laterally. This is medial, this is lateral. This is the TRN, this thin band of cells. And the stain here is for GABA. So when you see dark staining here, you have cell bodies and terminals that express GABA. So you're seeing inhibitory systems in the thalamus. And you see there's lots of GABAergic staining in the relay nuclei. Here's the LGN, as well as the thalamic reticular nucleus. So if we take a region like this and blow it up, this is what you see. Thalamic reticular nucleus has fairly large cells, and 100% of them are GABAergic. That's why they're stained dark here. But if you look in the main relay nuclei, we also have GABAergic cells. They're smaller, and these are the interneurons. That is, they don't project to cortex, but they provide local circuitry, and they innervate the relay cells. These are the larger cells here. They're unstained because they're not GABAergic. They don't use GABA as a neurotransmitter. They use glutamate. And what we have are two types of inhibitory neurons, and both of them concentrate their input onto the relay cells. Why we have two systems and what the difference between them isn't entirely clear, although there's some differences in anatomy that provide some insights. But one thing you can see here, if you can imagine, if these GABAergic cells are very active, they will so inhibit the relay cells that they can relay no information to cortex. The gateway through thalamus is closed. Conversely, if these cells themselves are inhibited and not firing very much, the gate is open and the relay to cortex is allowed. And you have the gate open or closed and various positions in between. Gating, we think, is one of the really important roles of thalamus, but there are many other roles as well, some of which I'll have a chance to talk about and others I won't. Okay, that's a very simple but straightforward, I think, function for thalamus. Now, if we put all this in a cartoon circuit, we see the following. This is a relay cell, and these are imaginary three relay nuclei. Could be LGN, somatosensory, auditory. And each of the TRN is also divided up into zones that reflect a relationship with each of the relay nuclei. So we have inhibitory input to the relay cells from these local interneurons. We also have inhibitory input from the thalamic reticular nucleus. The relay cells project up to cortex, but en route, they pass through the TRN and they provide collateral input, this excitatory, onto the TRN cells. We also have a feedback from cortex. This feedback that's shown here actually emanates from layer 6, the bottom layer of cortex. I'll have more to say about this. And as these axons come down, they branch. Individual axons branch to innervate relay cells and also the local interneurons. And these cells are glutamatergic, these cells are glutamatergic, these cells are GABergic. Those are the main cell types we have in thalamus. Now, to complete the circuitry of thalamus, there's another major input that comes from cells scattered throughout the brainstem. We call this the brainstem. Reticular formation has lots of names. I don't want to get into confusing terminology. But if we took a cross-section here, we'll see that these cells project very diffusely and widely to thalamus. Some of them reach cortex. And through that cross-section, these are the cells that would project to cortex. Just to give you an example, they're scattered throughout the brainstem. They don't form into very nice nuclei. So if we put all this into context and use the lateral geniculate nucleus as an example, this is the relay of retinal information in cortex. We see the retinal input to relay cells, excitatory input. This shows the transmitters used, the projection up to mostly middle layers of cortex. These are the showing the inhibitory input from the reticular cells and the intraneurons onto relay cells. This is the feedback from layer 6, branching axons that innervate the inhibitory cells as well as the relay cells. And what this tells you is that, because these are all excitatory synapses, is that this pathway can provide both direct excitation of relay cells, but also disynaptic inhibition. And so the net result on relay cells is quite difficult to understand, and we'll spend more time talking about this. On the other hand, the input from the brainstem reticular formation, also through branching axons, innervates all of these, but it does something very tricky. And if you'll notice here, it provides excitation of relay cells, but the same axons, using the same transmitter, mostly acetylcholine, but others can be involved, inhibits the inhibitory neurons. And that means that while these cells are active, you not only get direct excitation of relay cells, but you get a removal of inhibition by inhibiting the inhibitory cells. And so what you would imagine is that when these cells are active, the gate is going to be open. When these cells are inactive, the inhibitory interneurons are allowed to become activated. The relay cells are less depolarized, and so the gate would be closed. Is that clear to everybody? Okay. Question is how this is done. So this is the example of the retina innervating the LGN. The main difference between the LGN and any other thalamic nucleus is the input to be relayed. The other non-retinal inputs are pretty much conserved throughout thalamus and across species. There are differences, to be sure, between nuclei and between species, but I'm going to ignore those. So the first approximation, you have the same non-retinal innervation in the circuitry. Again, what's different is what's relayed. So if this were the somatosensory thalamus, this would be the medial lemniscus providing somatosensory information, and so on. So again, how does these cholinergic cells mainly do this trick? The answer is that they activate receptors that are postsynaptic that are quite different. And I'm going to take a little detour to talk about postsynaptic receptors because I think they're generally more important than appreciated both in thalamic circuitry and cortical circuitry, and I think this is an important issue for modelers to take up. There are two kinds of receptor, basically. One type is called an ionotropic receptor. The examples I'll use are the AMPA receptor and the GABA-A receptor. Now, for the AMPA receptor, when the neurotransmitter glutamate binds to the receptor, which is a molecule in the membrane, this is the outside the cell, this is inside the cell, you get a very fast conformational change that opens the pore, a sodium or calcium channel. It opens up, and this happens very quickly because the channel is as part of the protein that's affected by the transmitter. And once this opens, sodium or calcium in some cases can flow into the cell, so you get a net flow of positive ions into the cell because of the concentration gradient, And that means the cell becomes more depolarized inside with these positive ions, and you get a very fast EPSP. It's called excitatory postsynaptic potential as a result of this. The other main kind of ionotropic receptor is the GABA receptor. It's a very similar thing. GABA binds to the receptor. A chloride channel opens in this case, and now a negative ion goes inside the cell, and that's going to hyperpolarize it. Now this is all contrasted to what we call metabotropic receptors, and there are a bunch of them. We have them for glutamate, acetylcholine, noradrenaline, dopamine, and so on. And they mostly involve potassium channels. So in this example, where the potassium channel is open, we have a very complex receptor, much more protein, much more complicated than for ionotropic receptors, and it includes what's called a G protein. When the transmitter binds to the receptor, the G protein is released and swims through the membrane to interact with another protein called an effector protein. And this leads to an explosive chain of reactions that are called second messenger pathways. A bunch of biochemical reactions are set off. Some of them can affect many things in the cell besides ion channels. They can affect even gene expression. So when you activate metabotropic receptors, you get much more, possibly, than just changes in postsynaptic potentials. And that's not very well explored. Anyway, one of the things that happens is, in this case, a potassium channel that's open, allowing potassium to leak out of the cell, is now closed. And this prevents the leakage of potassium, allowing a buildup of potassium ions that otherwise wouldn't be there, and that's a depolarization. But notice the time course. This is a very fast time course. These depolarizations can last hundreds of milliseconds to several seconds, so it's a very slow, prolonged process. And this is what we get for a type of metabotropic glutamate receptor. It's called group 1, and also the M1 type of muscarinic receptor that is used for acetylcholine. We have the opposite as well. Metabotropic receptors that, when activated, open the potassium channel. Now these positive ions will flow out of the cell, and that leads to hyperpolarization because you get less positivity inside the cell with the same kind of time course. This we see for group 2 metabotropic glutamate receptors, the M2 type of muscarinic receptor for acetylcholine, and the GABA-B receptor. Notice one point I want to emphasize is that glutamate, when it encounters this kind of receptor, can hyperpolarize. So glutamate can be inhibitory, and we see that a lot in cortex in particular. Most people think of cortical inhibition as only GABAergic, but a lot of it can be glutamatergic as well. Okay, so one or two other things about these receptors that are important. The ionotropic receptor, because it has a direct action and is very brief, these receptors are found right at the synapse. And these are examples of them, the AMPA and NMDA receptors leading to EPSPs. Acetylcholine has a nicotinic receptor that's ionotropic, leads to an EPSP. And GABA has a GABA receptor that produces an IPSP. The metabotropic receptors seem to be located further away from the synapse, forming an annulus around it. And I'll get back to why that might be important. This operates through the second messenger system, so it's indirect. It produces prolonged EPSPs or IPSPs that last hundreds of milliseconds to several seconds. Glutamate activates group 1 metabotropic receptors to produce EPSPs, or group 2 for IPSPs. Acetylcholine also can do EPSPs or IPSPs depending on the receptor. And the GABA-B receptor gives you a long IPSP. The location here, yeah.
Glutamate and GABA, one of them is excitatory, one of them is inhibitory?
Glutamate can be either,
okay,
depending on whether the receptor postsynaptic is group 1 or group 2. GABA, as far as we know, is always inhibitory.
thank you.
What is the proportion of excitatory glutamate receptors versus inhibitory?
Great question. I wish we knew. So what I can tell you, I don't think I have a slide that shows this, is you can use immunocytochemistry to see the extent to which you get staining of these kinds of receptors in thalamus and cortex. And in cortex in particular, you see a lot of both types. But, and most glutamatergic inputs in cortex and thalamus activate one of these. But I don't think we know enough to know what the relative numbers here are and the relative strength. So you get both kinds.
Is there an approximation of like there's definitely more of group 1 versus group 2, or
We don't know.
really?
If I were to guess, I'd say more group 1 than group 2 based on what we've seen, but it's not a very quantitative approach. Certainly get a lot of both.
Okay.
Because of the location of the receptors, this tells us something about the sensitivity of these receptors to activation of the presynaptic axon. So because these ionotropic receptors are right at the synapse, any frequency of firing here, even a single action potential can activate them. So when this axon is just firing at a very low rate or a very high rate, you're going to get ionotropic responses. But because of the lateral location of the metabotropic receptors, we think this is the reason, you don't start activating them until this axon starts firing at a relatively higher rate, roughly 10 hertz or so. Because you need to eject more transmitter, it takes more transmitter released here to get to these receptors. We think that's the reason for this. But this is interesting because it means that when you have postsynaptic receptors of both types, only the ionotropic receptors are going to be activated when when this input is not doing much. But when the input is doing something really important and gets excited, then you bring in the metabotropic responses. Any questions about this so far? Okay. So if we go back, just to review very quickly, the muscarinic receptors for acetylcholine, the M1 receptors produces an EPSP, the M2, an IPSP, and a prolonged one at that. And so if we go back to this circuit, the axon branch that innervates the relay cells from these cholinergic inputs activates M1 receptors, and that gives you the EPSP on the relay cells. But the branches carrying the same transmitter that innervate the inhibitory cells, the GABAergic cells, activate M2 receptors, leading to IPSPs and shutting down these cells. And that's how this input does that nice trick. And again, it means when these cells are active, the gate is open. When these cells are inactive, the gate is relatively closed. Here's a nice example of this from a paper from Stereon many, many years ago. I forget what year this was published, but at least 30 years old. What he's done here is he's taken a cat and he's stuck an electrode, a gross electrode, into the LGN, the visual relay, and recorded the response to electrically stimulating the optic nerves. And what you see here is this complex waveform when you record with this crude electrode, and I'll take you through it. This first wave is the presynaptic action potentials coming into the thalamus. This is the postsynaptic response. And you can tell whether the cat is alert, sleeping, or drowsy by measuring the EEG at the same time. You notice when the cat is alert, there's a strong postsynaptic response, the gate is open. When the cat is asleep, the gate is closed. There's no effect on the retinal output because the retina is not influenced by levels of alertness. When the animal's drowsy, you get a more variable response. And what we know is that those brainstem cells are very active during alert behavior and are shut down during sleep, and during drowsy periods they are intermittent. Yeah.
Should one think of this BRF as really being a universal gate where everything is closed down and shut down?
No, I'm generalizing.
More selective?
It's more complicated. I'm generalizing, and I'm only talking about the cholinergic effects, which we know best. There are noradrenergic, some dopaminergic inputs, serotonergic, so there are a lot of different transmitter systems used, and which ones are active in a particular nucleus varies. So the LGN, for the sensory ones, seem to be dominated by cholinergic input, which I'm talking about here. There's range for a lot more variable effects that aren't really very well explored. Most of what we know about thalamus comes from studies of the visual thalamus. And when it comes to this kind of thing, it's tempting to extrapolate, but we really don't have the evidence that this is true for all thalamic nuclei. But something like this is very likely. Any other questions? Okay. Now I'm going to switch to cortex. Again, this is very superficial. There are actually three kinds of cortex. Paleocortex, which is the olfactory cortex. Archie cortex, which is a hippocampal formation. I'm going to talk only about neocortex. These other cortical areas I don't really have time to go into. And most of the studies that any of you will be familiar with are really mostly concentrating on the neocortex. This is the names imply that this is the newest part of our cortex to evolve, the most recently evolved. Cell types. We've heard about cell types before. If you're a splitter, there are probably as many as 50 different cell types in cortex. To simplify things, I'm going to lump things horribly and talk about only 4 cell types. But you have to just keep in mind that each of these cell types I'm talking about has lots of subclasses. The most prominent cell in cortex you've already heard a lot about. This is the pyramidal cell. This is shown so that the top of cortex would be up here, the bottom is down here. The pyramidal cell is so called because its cell body is shaped like a pyramid, roughly, with the apex pointing up towards the surface of the cortex. It has basal dendrites and apical dendrites, and these apical dendrites can reach the cortical surface with lots of spines on them. The pyramidal cell, some of the pyramidal cells, not all of them, represent the main output of cortex, the main efferents. So these axons will project into the white matter, project other cortical areas, and in some cases project subcortically. But it's important to keep in mind that they also provide a lot of local innervation through these collaterals. So they're not just efferent cells. Interneurons come in 2 shapes. We're calling them stellate cells, or smooth, and there's spiny, as the names imply. These are thought to be exclusively inhibitory interneurons, and these are excitatory interneurons. Of course, these and particularly the inhibitory ones come in many exotic shapes, but these are by calling them interneurons, they don't have axons that project out of cortex to subcortical structures, and very rarely very far within cortex. The final cell type, which I won't spend much time talking about, it's called a fusiform cell. It has a bipolar dendritic tree. This also has axons that can leave cortex, as well as the pyramidal cell. And the orientation of the bipolar dendritic tree can be any orientation. This is vertical, it could be horizontal, it could be anywhere in between. So these are the 4 main cell types. Again, each of them have lots of variations that I'm not going to go into. How do they fit with layers? Well, I think you all know that we have layers of cortex, and what you see here are 3 cartoon examples. These represent Golgi impregnations. You've heard about the Golgi stain that Cahal made famous. This stain randomly picks out a very small percentage of cells and stains them entirely. So you get the cell body, the dendrite, the axon. If it stained every cell, this would just be a black mass. So it's a nice thing. It randomly picks out cells, and we have no idea what the rationale for cells it picks out is. This is a Nissl stain that stains every cell body, every neuron, but not much of the dendrites and not the axon. So you can see where all the cells are. This is a myelin stain that stains myelinated fibers. Now, these cells are arranged in layers. The top layer is called the molecular plexiform mesonual layer, layer 1. It has very few cells in it. It's mostly a synaptic region where synapses are formed between apical dendrites of these pyramidal cells and fibers running in layer 1, which mostly come from other cortical areas, but some from matrix cells in thalamus. Layer 2 is called the external granular layer because it's comprised mostly of granule cells, or stellate cells. Layer 3 is the external pyramidal layer, comprised mostly of pyramidal cells. We have the internal granular layer, or internal stellate cell layer. We have the internal pyramidal layer. These are where the largest pyramidal cells are found, and this is the layer that provides much of the input out of cortex to subcortical structures. And finally, layer 6, which is a multiform layer, having a lot of these unusual cells that are bipolar type cells. But even though I said that this layers 2 and 4 are mostly stellate and 3 and 5 are mostly pyramidal, all cell types are found in all layers except layer 1. So you have pyramidal cells in layers 2 and 4, you have stellate cells in layers 3 and 5. But layers 2 and 4 are mostly stellate, 3 and 5 are mostly pyramidal. Now, early anatomists in the 30s, looking at material like this, immediately saw what turns out to be important, especially when you look at the initial stains, the cells seem to be arranged in columns with gaps between. And those gaps are filled by myelinated axons running up and down the column. And columnar organization of cortex, which was first described by Mountcastle and later demonstrated more convincingly by Hubel and Wiesel, turns out to be an important feature of cortex, we believe a general canonical feature. And this is an example from a paper by Hubel and Wiesel that shows what the columns mean. These columns are roughly 0.5 millimeter across. And what they're showing here is they put an electrode through the visual cortex, and this electrode encountered single cells, and each cell they plotted the receptive field for. What they found that the receptive fields of the cells, as long as you stayed within a column, fell on top of each other, so they were representing the same point of visual space. But more interestingly, they had the same orientation selectivity, let's say horizontal in this case. If you stayed within a column, all the cells were interested in the same orientation. If you cut across columns, you saw that the orientation specificity of the cells in the column shifted gradually around the clock. And their point was that each column takes a part of visual space in this example and is interested in the main features, a particular feature that it is involved in analyzing, in this case orientation. There are columns, we think, for direction, for spatial frequency, for all sorts of things. So the columnar organization.
Where does the input come in? Where does the output go? And where's layer one? Where's
You're
layer
getting way ahead of me. Okay, I'll get to that. I'll get to that soon, I think. So the columnar organization, again, is a key feature of cortex, and we think that if we understand how a column functions, that we will understand a great insight into cortical functioning. I will say that the connections within a column are mostly up and down the column. You have relatively few connections running horizontally in cortex. Okay, to answer your question, let's look at the inputs to a cortical column. From other cortical areas, they terminate mostly in layer 2. From thalamus, core regions, mostly in layer 4, but also some in layer 6. The matrix to layer 1. Claustrum, which is a structure I'm really not going to say much about it. It's a thin layer of cells that sits below cortex, innervates layer 6, but ignore that. We get lots of modulatory inputs that very diffusely innervate cortex and innervate pretty much all layers. The basal forebrain, which provides a lot of this, provides cholinergic input to all of these layers. Now for the outputs, if you want to send information in another cortical area, it comes mostly out of layer 3. Layer 5 provides the main subcortical projection to many sites, which includes thalamus, also spinal cord, almost any brainstem nucleus that is going to get an input from cortex, it will get it from layer 5. And layer 6 also provides input to thalamus. And I'll talk about the difference between these two inputs to thalamus. And then we get claustrum also. So claustrum forms this loop that is not well understood, and I'm not going to have any more to say about it. These laminar patterns are meant to be a simplification, and they represent the main tendencies for the relationships of layers to inputs and outputs. But there are exceptions. For instance, thalamus probably innervates all layers, and you can get outputs from to other cortical areas from other layers as well. But what's interesting about this generalization is that the upper layers, layers 2 and 3 in particular, are interested in communicating with other cortical areas, getting its input here and its output there. The bottom layers are mostly communicating with subcortical sites. And that's a generalization we can make about cortex. So this is how the layering generally applies to inputs and outputs. And if we consider a single column, we'll start in layer 4 because arbitrarily, because this is where most thalamic inputs come in. Layer 4 cells project, all this is going to be within a column up to layers 2, 3 mostly. You go from layers 2, 3 down to layer 5, also down to layer 6. From layer 5, you also get down to layer 6, and from layer 6, you close the loop and get back up to layer 4. Imagine all this is within a column. And again, this is a simplification, but it's a useful one to start thinking about how cortical columns are organized. Yeah,
So the outputs from the...
you have to speak up.
The outputs from a single column. How many columns do they usually span?
Well, each column is about 0.5 millimeter across, so depending on what the area of cortex is, the surface area, you can roughly figure out how many columns that area will have. And the size of a column, we don't have a lot of data on this, but the size of a column is pretty much fixed throughout cortex, we think. So a larger cortical area will have more columns, and a smaller one will have fewer columns.
So sorry, and a single column can talk to something quite far away.
A single column,
Can have outputs going...
yeah, right. So if we go, let me go back to the previous slide. So in this example, a single column, this cell is in a column, can project to other cortical areas very, very far away, including to the other side, the other hemisphere. And the same for the input. So they can communicate over quite a large distance. Yeah.
So in the data you showed about the LGN in the cat, how do we know that that modulation was due to input from the thalamus?
How do I know?
That the modulation was due to input from the thalamus.
I'm sorry.
So in the data in the cat, in the LGN in the cat,
Yeah.
how do we know that that modulation of the awake and the sleep was
Oh.
due to input from the thalamus?
It was input from the stem, I believe.
Yeah. So
Sorry, could you say that again?
if we talk about that slide I showed that the input through LGN was being shut down,
Yeah.
we think that's mostly because of activity in the brainstem inputs to thalamus that are correlated to whether the animal's awake or asleep. So let me go through that again very quickly. If the animal's awake, the cholinergic inputs from brainstem are very active, and we know that. They will excite relay cells and inhibit their inhibitory inputs, disinhibit them. So they're both exciting and disinhibiting them, which makes them very responsive to visual input. The gate is open. When those cells shut down, and they shut down when the animal goes to sleep, you remove direct excitation of relay cells, and you also remove the disinhibition. You allow the inhibition to take over. The relay cells are relatively hyperpolarized, and they don't respond to the retinal input anymore. So we think that's the simple answer to that question.
But given that there's so much going on, couldn't it be some other pathways as well?
Yes, we can't rule that out, but let's just say that that's our understanding at present for how this is working. There is input, the input from cortex can also have an effect on this, but I'm talking about overall general effects to shut things down. You can shut things, and that would be a global non-topographic effect. The layer 6 input, and I hope to get to this, can do this in a highly topographic manner. Maybe that's what you're asking about. So you can, and what I've talked about was opening or closing the gates throughout thalamus in a very global manner. But there are pathways that allow you to do it very specifically for single cells even, small groups of relay cells. And I hope to get to that. Okay. Areal specializations now. I talked about a general cortical type, but actually there are many types of cortical areas. And this shows, a lumping approach that only has 5 types. Type 3 is the general area of cortex that has all 6 layers very clearly demonstrated. Type 5 is called granular cortex because the granular layers, 2 and 4, are highly represented, lots of stellate cells, not very many pyramidal cells. And we see that associated with primary sensory areas, the visual cortex, primary visual cortex, auditory cortex, somatosensory cortex. The other end, we have agranular cortex, very few stellate cells, but lots of pyramidal cells. And this is related to motor areas of cortex, because you have lots of pyramidal cells, lots of outputs. And we have types in between, and there have been attempts to map this out. So there are variations, and in some cases, it's hard to see all the layers. In the case of purely sensory or motor cortex, for instance, here it's hard to see where layer 4 is. But if you use special staining techniques, you can demonstrate that all these layers actually do exist. They're just harder to recognize. Now, in the 19th, yeah.
So just
question, now that we have the whole cortex up here, so are columns across all the cortex? Does every local area have columns?
Columns are found, we think they're found throughout cortex, but most of our information about columns comes from sensory cortex.
Yeah, so I was going to say, I've seen this in the input areas and the, so I guess like with whiskers for rodents and with V1, there's a lot of studies about columns. So do columns also exist in, say, hippocampus?
In hippocampus?
Yeah.
No, I don't believe so.
Okay.
This is a neocortical property. In the 19th century, there was a cottage industry among anatomists, largely German anatomists, to see how many different kinds of cortex they could define just by histochemistry. And some of them came up with over 200, and it's an historical interest to some. But the important map of cortex of all the cortical areas that has survived today is called the Broadband Map. And it's important only because the numbers that are used are still applied today. And so Brodmann defined 50 some odd areas based on these slight differences in lamination. Area 17 is striate cortex, primary visual cortex. 41 and 42 would be the primary auditory cortical areas. And we have motor and somatosensory areas as well. The only reason why this is at all important is because those numbers are often used. And you might want to understand that when someone says they're looking at responses in area 17, you should understand where those numbers come from. In some cases, like area 17, the correlation between the structure and the function is pretty clear, but a lot of these areas that Brodmann defined really don't seem to have any specific correlate to a functional area. So its use today is not very significant except for some of these areas, particularly that define the primary motor and sensory areas. It's also valuable for neurosurgeons if they say, I found a tumor in area 46, it helps identify to a colleague where that tumor is. But otherwise, these maps are mostly of historical interest. In the old days, before the 70s, we thought there was primary sensory cortex. Let me go back a bit. Primary sensory cortices, some primary motor cortex, and everything in between was called association cortex because we didn't know what it was. It turns out there's probably very little association cortex at all because we now know from mapping studies that started in the 70s and continue today that the back half of cortex is almost all purely visual. We have lots of somatomotor and auditory areas. And this is a very old map taken from the 1970s, I believe. As more areas are studied with more sophisticated techniques, we find that most of these areas of cortex have a relatively specific sensory or motor function. The main areas that could be called association would be frontal areas that don't seem to have that kind of sensory or motor function. And this is a map, a very useful map that's very important from David Van Essen and colleagues. They took a monkey's brain and with computer and help, they unfolded it. And so this would be the lateral surface, the medial surface, and this would be the unfolded, and this is V1, or area 17. What's interesting, when you look at the map of a monkey, or and this also would apply to the human, is that the back half is almost all visual. There are 30 some odd visual areas. Here's the auditory regions. Here's the somatomotor regions. So most of the cortex, and these maps again are about 30 years old, most of the cortex have fairly specific sensory or motor function. In the case of the visual areas, it's believed that there are, this would be another way of looking at it, the connections, again from David Van Essen's lab, go through Thalamus. And they arranged all these areas into hierarchies. The idea is that information is flowing up these hierarchies, and they identified 5 or 6 parallel hierarchies with 5 or 6 different levels. It's been a very influential map. I'm going to make some modifications of it if I have time. One thing that's interesting is the pulvinar, which is a very large thalamic nucleus in this scheme, has this sort of arrows pointing up for kind of with I could put question marks here, but I'm going to try to put this into the scheme more firmly. So this is still pretty much our current view of cortical processing, particularly in sensory areas like visual cortex, that it's hierarchical, and you go from basic information analysis to more and more complex, sophisticated things up these several streams.
Are there some nice online data sets of these maps?
Sorry?
Are there some nice online data sets of these maps?
Yeah,
I think
Bruno's been involved in this, so he would
David
be.
Van Essen has, they do this all that map I think was actually done by hand. But the ones they do now by computer, he has a lot of them online available. Yeah. And also I think maybe the Allen Institute does as well.
Right, that's what I was also.
So we have a connectivity atlas for mouse brain, which contains information about connectivity between areas.
Okay, thank you.
Yeah, okay, so I'm going to switch gears now. If there are any questions up to this point, I'm going to talk about the layer 6 feedback. Yeah.
How does it differ between humans and more primitive?
That's a great question. How do we relate all this to humans? A lot of this work is done at a wide range of animals, including some humans. We go from mice through carnivores like cats and ferrets to monkeys, and then humans. And by comparing these different species, we try to get a sense of what are general properties of the mammalian cortex. And those will apply to humans, as opposed to things that are unique to each species. You try to sort that out by looking at information on lots of species. One of the problems today is that when I started out many years ago, the species that were studied, there must have been a dozen different species, which gave you a pretty good platform for trying to understand what is general for mammals and what is not. Now we're down to two, monkeys and mice, and they're very far apart. So we're having trouble now figuring out what's general, what applies to mammals, and therefore to humans. We believe, I think this is correct, that what I've been talking about so far and what I will continue to talk about is an attempt to pick out the properties that are general mammalian properties that will apply to humans. But we're not on very firm footing because we just don't have enough comparative data. And the human data is largely, I should say, limited to imaging like MRI. You can't do these kinds of experiments, obviously, in humans to get those kind of data. So again, we think this is, what I'm talking about is related to humans, but that's a big problem. Yeah.
Where is the information from the different sensory areas fused?
That's a good question, too. We used to be thought in the old days that's what association cortex was doing. But there are areas that have been identified that are multisensory, and this is where it's happening. The superior colliculus, which is a subcortical structure, is an important structure for this, and is a very old structure, and is an area that is used for that in non-mammalian vertebrates like birds and reptiles and so on. But, and that's still true in mammals. But there are cortical areas that have been identified that are multisensory, and I think that's an important point because most of us who study sensory systems study a single system. We concentrate on the visual system or the somatosensory system and so on, but that's not the way our brain works. We're using multisensory integration all the time, and you think about it, imagine you see a flock of birds flying overhead. If you just had vision, you might not know what they were, but you listen to them at the same time and you hear whether they honk or they whatever abduct us, quack. And by putting multisensory information together, you immediately identify what you're dealing with. And this is what we do all the time, but people don't study that very much. There's very few labs that actually study multisensory integration. Okay, I want to talk now about this layer 6 feedback. And you see one of the problems here is that the feedback can either directly excite or indirectly inhibit. And if this were the circuit, you'd wonder what the heck is going on with relay cells. When these cells are active, you do both, and that would seem to not be very interesting. So here's what the circuit could look like. We're going to do it at a single cell level. It will directly excite this relay cell and indirectly inhibit it. And the change in membrane potential here, if these were balanced, would be 0. And you might think that was a useless thing. But work done by at Larry Abbott's lab, Chance et al. Paper some time ago, showed that when you have balanced activity, it doesn't have to be balanced, but even if it were balanced, even though you're not changing membrane potential, you're changing the input resistance of the cell and other factors that affect the gain of synaptic transmission of other inputs. So if this were a geniculate cell, the retinal input, the gain of the retinogeniculate synapse, the size of the EPSP, would be greatly reduced if this circuit were activated. So it's a convenient, useful way to control gain of synaptic transmission. Having shown this, this is a different kind of circuit using the same elements, and actually we have evidence from a number of labs, including ours, that suggests that this is actually not just common, but maybe the dominant way things are organized. And you see how different it is. When this cell is active, you don't have balanced inhibition and excitation going in all cells. Some cells are excited, and some cells are inhibited. You see how different these two circuits are. Okay, and again, we think this is more common, but both probably exist. This is not a homogeneous pathway. But there's a problem here, and I'm going to try to, I have a difficult time coming up with a way to explain this, so please ask questions if it's not clear. Let's imagine that the circuit is really based like this, and we have lots of cells that are doing this at the single cell level. When any one of these cells is active, some relay cells will be excited, others will be inhibited. But the techniques we use to study this pathway are limited because we don't activate single cells and record from single cells. Instead, we excite or inhibit whole groups. Now, if you look at the way this is organized, if you were to excite all of these, even with modern techniques that use things like channelrhodopsin, the end result is that all of these cells will be excited and inhibited. Is that clear to everybody? Even though at the single cell level, it's all going to look like this, when in fact, if you had pattern activation, spatial pattern, you get a very different response down here. And this is an important proviso for most experiments that are being done. When we talk about knocking out a pathway through lesions or chemical ways of reducing all these cells or activating them, even with modern techniques, we're not seeing the actual way the circuit is behaving. And it's for that reason, I think, that it's very difficult to get clear evidence for what the effect of this pathway is on relay cells. Because all the experiments that I know of, with very few exceptions, either excite or inhibit large groups of these cells, and you wipe out the beautiful, detailed single-cell circuitry that is really playing the role. Question?
Yeah, sort of, if you record in layer 6, you do see pattern activity, right? You do see individual cells active and lots of other cells not active.
Yeah.
So it is more natural.
Of
Like
course.
you could do that with a...
Right, so you can imagine if you had, these cells have very discrete receptive fields. So if you were exciting this with a receptive field, that same stimulus would likely inhibit the neighboring cells because there's a lot of this direct excitation, and when you get outside the center of the receptive field, you get other kinds of effects. And so there's every reason to believe that the way this would respond to visual stimuli would require this kind of detailed single-cell circuitry. But again, that's completely wiped out when you do large-scale excitation or inhibition. Yeah.
Anything known about how much of this wiring is genetic and how much is learned?
No. There's actually very little about the cortical input to thalamus in terms of development. The retinal input has been studied quite a bit, and there it seems to be, and this was described, I think, by Jeff, that you get an exuberant initial connection pattern that is pruned back. I don't think we know very much about how this develops and how much is genetic. Okay, no other questions about this? I think this is an important qualification for interpreting many studies that are done today where it doesn't just apply to this system. Imagine that other systems are organized in this cell-specific way. If you do massive inhibition or excitation of a pathway, even with modern techniques, you run the risk of obliterating the patterns that are important. One last thing I want to say about this layer 6 input. So these layer 6 cells project down the thalamus, and they can have a pretty powerful effect because they, as you can imagine, they can excite the relay cells and they can influence the local inhibitory cells. And in answer to a question I think that was asked earlier, this is a way that the cortex can very locally control gating, controlling some relay cells by inhibiting them and exciting other relay cells. But another important aspect of these layer 6 cells that is only just recently becoming realized is that a branch of their axon goes up into layer 4 to innervate the target cells of thalamus, which means these layer 6 cells are pretty interesting because not only do they affect thalamocortical transmission at its source, but also at its target. So it can affect it at both places. And one thing I'll just throw out there, this input, which is glutamatergic, activates mostly group 2 metabotropic glutamate receptors here, which would inhibit the cell. And so you'd think that this is a glutamatergic pathway that would excite the cell. In most cases, it's the opposite.
Just to be clear, that last dot dashed arrow going to layer 4 cell, that's coming from the layer 6 cell?
Yeah.
Is that what you meant to draw up?
It's an axon collateral.
Collateral.
Same cells. Yeah.
Just to get a sense for approximately how many cells are there, well, let's say in a cortical column or like per layer?
Oh, those numbers are known. I can't tell you off the top of my head. What I can tell you is for every retinal axon coming in here, there's about 100 of these going down. I don't know if that helps. The numbers are known. There's a literature on it. I don't remember the numbers off. Maybe somebody in the audience does.
And they all sort of function sort of together. They have a similar function because they all have the same receptive field.
Well, they have similar receptive fields within the same column. But there's more interest in these layer 6 cells, and it turns out that there are at least 3 different types of layer 6 feedback. And if I have a chance in the next hour getting into more complicated stuff, I'll bring that up again. So there's heterogeneity here. It's not homogeneous. Certain properties are the same among these cells, but they are different, and they have specific kinds of targeting of specific kinds of relay cells. So there's a lot of specificity here that's been overlooked.
Okay.
I'm going to stop here. Any other questions? Yeah.
You've shown here the inhibitory signal inhibiting the input coming from thalamus that goes back to the same column. Is that the most common case, or is?
Yeah, so that's a good question. I meant to say this and I haven't. This circuit represents from cortex to thalamus a feedback. We call it a feedback because the column that's innervated by the relay cell is the column in which these layer 6 cells are located. So this is a classic feedback kind of loop. Yeah.
Are you going to talk at all about the temporal dynamics? Because it seems that excitation, inhibition, disynaptic, etc. This is a good way to generate oscillations and things. So.
No, I won't. I don't plan to get into that. It's a very interesting topic. It's an important topic, but at this level, there's not much I can say about that. Yeah.
So just to the previous question, as far as I know, a single column would be something like 30,000, maybe 50,000 cells in a cat.
Okay.
So tens of thousands of cells across them.
And I might add that in layer 6, depending on the species, it's no more than 50% of the cells are doing this. Other layer 6 cells are projecting to other cortical areas. So this isn't just layer 6. Yeah.
So getting back to something I wanted to know, I've been told by some people that the input to the cortex, the visual input, it goes to layer 1. I've been told by others, layer 3.
No. The main, if we're talking about visual cortex, from the lateral geniculate nucleus,
yes.
The retinal relay, it's going mostly to layer 4 with some to layer 6. The inputs to, this is the core projection. I'm going to get to this in the second hour, I hope. There are some cells in the LGN that are matrix that project mostly to layer 1. In the cat, those are the W cells, and the monkey, those are the coniocellular cells. So that pathway does exist, but as I try to say in the beginning, we don't know much about that at all. That's very poorly understood what its function is. I think we understand a lot more about the projection to cortex that's terminating mostly in layer 4.
Mark?
Yeah.
So it's a lovely presentation, really. But I'm just wondering, a lot of the evidence would be that the vision uses polling, so the cortex is constantly asking questions of the thalamus and what's out there. And in that regard, for say whisking, which is in vision, but the idea is that the thalamus plays a role in phase locking just to read the stimulus. And so the actual question is, maybe we have to worry about what's going on to make sense of the very rich anatomical structures in there. If we don't ask these functional questions, we're going to be held back.
Right, so maybe we can put that question aside until I talk about the role of thalamus in cortical pathways, because I think that's where a lot of this possibility comes up. But it's going to be mostly speculation. Quick time to take a break.
Yeah, one more question. I'd like to get my question in. Can you say anything about the relation between these layers and the layers in the neural networks that are used, for example?
No, I can't. Easy answer, I don't know.
This is all mammalian cortex. How does it differ with
Zebrafish don't have neocortex.
Yeah, I mean, do they differ
So that's